So welcome back to the second case study for micro remains. And in this case study, I will present the joint results of microbotanical, so phytoliths, and macrobotanical seeds and fruit analysis. The latter was conducted by Doa Karakaya on the remains from a Middle Bronze Age layer at Zincirlikuyu. The basis of this work was laid through a multidisciplinary study of seeds and fruits, charcoal, phytoliths, diatoms, dung spherulites, and the faunal material, which focus on the environmental conditions during the Middle Bronze Age and will be published soon. Today I want to highlight the potential micro and macro botanical remains can offer by focusing on the different benefits of each field by combining the sometimes at the first glance contradicting results with the archaeological data. What I will be talking about come from the DD1 building complex, which is located in an area south of the so-called Hinani 1 and south of another building complex, DD2. The DD1 building consists of 3 rooms, DD1, 2, and 3. Room DD1 contained a storage jar of bitter wedge, while in room 2, 40 spherical pilgrim bottles were found, probably intended for transporting wine, as well as pressing and grinding equipment. In room DD3, perforated vessels and vessels standing along one wall indicate the handling of liquids, possibly wine. Although room 2 and 3 show evidence of food preparation, no hearths or ovens were found. A microfossil pilot study was undertaken on 19 samples from 9 different loci. These can be assigned to rooms 1, 2, and 3 and to an outside area. The samples derive from sediments designated for macrobotanical study and were separated from them prior to flotation. The analysis of diatoms, more precisely the glass cell wall remains of Algiers, were included in the course of the phytolith counting in the respective slides. Furthermore, samples of dung spherulites were prepared. For the current study, 105 archaeobotanical samples from room 1, 2, and 3 were analyzed to understand the use of plant resources in this complex and to gain an understanding of the land use. Start with room DD1, the room with a storage vessel full of bitter wedge. This deposit included over 38,000 grains, mostly bitter wedge grains, but 2% of the whole assemblage contained arable weeds. The absolute amount of phytoliths from samples from this room is generally high, and both free threshing wheat and barley could be identified from these samples. A sample from the outdoor area near a fruit processing installation contained, as expected, a high proportion of grass phytoliths and no multicells. So most probably there were activities connected with cereals and physical impact like grinding activity or at least with grinded products. Next context room is DD2. It is another room full of restorable vessels. The botanical contents of these vessels and kitchen tools were examined. The vessels in this room were mostly empty. Only one of them contained a low number of water-loving plants. In general, the deposits were possibly mixed during the destruction, which leaves little place for conclusive interpretation. However, the floor level of this room held concentrated finds of barley and lentil, but the exact provenance is unknown due to their displaced nature. The crop was deposited in the final hand-sorting stage and was broken either due to destruction or maybe for bulgur making. Lentil remains were collected in several loci, most probably indicating that the stored product fell down and spread over the room. For this reason, it is difficult to know the species composition of stored product, when brought to the settlement. It seems to be mixed with neighboring deposits. Only 2 samples were analyzed at the moment for phytoliths, and the results are very different, so nothing striking was observed. Next was room DD3, which contained the most interesting results in terms of plant deposition in an archaeological context. 3 loci in room 3 delivered good archaeobotanical information. The first one is a roof collapse at the northwest of the trench. The deposition in room 3 interestingly favors the small seeded grasses and other small wild plants rather than bigger ones. The reason of this bias would be related to the destruction impact of a wall collapse. Secondly, the flowering times of several wild plants coincides to summer or early autumn with a substantial amount of freshwater plants. In a wall collapse locus, larger sized seeds were found fragmented, but they could still be determined, for example, lentil, bitter wedge, and garden pea. Also cereals like wheat and barley, typical weeds and freshwater plants. Another locus demonstrated evidence of overburned animal bones and mollusk shells in addition of the several highly fragmented and burned cultivated cereals. Some other samples from the same locus contained lots of grape remains. From the point of view of phytolith analysis, the samples from room 3 are particularly interesting. Without going into detail, they provide, on the one hand, much more room for interpretation due to the number of samples from different contexts, and on the other hand, they show many identifiable morphologies. To sum up, a high proportion of inflorescence is found in the samples from Zingerli, especially in room DD3, while a certain proportion of woody plants comes from the rooms 1 and 2. It is noticeable that in the woody plants, the proportion of wood bark is almost always significantly higher than the proportion of leaves, which are present only to a small extent. Among them, the cultivated cereals, wheat and barley, with a high occurrence of barley, could be detected. Other plant family species are acid grass, are beech, possibly kermes oak, sweetgrass, and reed. 9 of the samples examined contained diatoms. Based on the description of the characteristics of the identified diatoms, all species seem to occur in flowing and stagnant water, with the most frequently identified species also occurring in brackish water. Dung spherulites generally occurred in very low numbers in the samples examined, with only the sample from the outer area showing a slightly higher number. While this generally indicates the presence of herbivorous animals, or at least their dung, in the settlement, the DD1 complex appears to have been generally kept clean. For example, there were no dung-related activities such as animal husbandry, but also the use of dung as a secondary product, such as heating material, which is confirmed by the absence of fire installations in the archaeological record, but also the use in architecture, for example, as an admixture in the mud bricks or in the plaster, does not seem to have been the case. Looking to the cultivated cereals, it seems that there are some distinctions between the rooms. While room 1 is devoid of barley completely, free threshing wheat is completely absent in room 2. On the other hand, both cereals appear in room 3 with high frequencies. However, the phytolith analysis show partially different results. Remains of barley and free threshing wheat can be found in all rooms, but they show up in room 3 with a higher proportion of barley. According to the results of phytolith analysis, cereal processing would be most likely in room 1, whereas the contexts in room 2 and 3 have a noticeable higher proportion of woody plants. We can state the following as preliminary results. Room 1 provided firm results on the composition of bitter wedge field around Zingerli. There are very few grasses in this deposit, but a high concentration of cereal phytoliths in room 1. In room 2, on the other hand, we encountered more mixed deposits. The vessels here were empty, but it seems that stored grains in some of these vessels were spilled to the ground. The few and non-specific samples for phytolith analysis cannot contribute to this. The assemblages in room 3 were very diversified, with several summer-autumn flowering plants. This could be related to the fact that most of our samples are coming either from a wall or from a roof collapse. The construction technique of both structures included grasses as tampering agent. Therefore, the presence of grasses, inflorescences, and several grains could be related to this factor. The very good conservation and the diversity of the identified morphotypes in the phytolith analysis also confirm this. It turns out for our first work on the samples from Sinjelehoek that integrating the phytoliths and seed data into our analysis could produce novel results in interpreting the plant evidence. However, it also shows the importance of the sampling strategy, which should be worked out individually for each method and taken into account in the field work. Thank you.